Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Nagar, Telangana State. We were discussing about respiration in the last class. We will have a quick glance so far what we have learned. We saw the definition of respiration, that is to keep all the metabolic activities in motion. The chemical energy that is stored in the organic compounds, which are formed as a result of photosynthesis, is released and liberated by the oxidation of the reserve food materials. So this process is known as the respiration, and it is a catabolic process where breakdown of these organic molecules takes place, liberating carbon dioxide and water. Okay, we saw the history of the respiration. Then we also see comparison of respiration and photosynthesis. See here, once again, you can see here in respiration, the respiratory substrate in the presence of oxygen, it undergoes complete oxidation, liberating carbon dioxide and water. And in this process, a lot of energy is liberated. Some of the energy is wasted in the form of heat also. So this reaction is known as exothermic reaction, as the heat is liberated. Okay, and it is a catabolic process as the larger molecules are break down into smaller molecules. And here the potential energy is converted into chemical kinetic energy. Okay? Potential energy is converted to kinetic energy. And here you can see here inorganic compounds combine to form glucose molecule, that is this carbon from the carbon dioxide is reduced to glucose, and it is a anabolic process and endothermic process where energy is observed. Okay, so it occurs in the presence of light and it occurs throughout the day. It doesn't depend upon light. Okay, so here respiration occurs in cytoplasm and mitochondria, whereas photosynthesis occurs in chlorophyll. So all these things we saw yesterday, and also seeing the structure of the respiratory apparatus, that is mitochondria. We said that mitochondria are also known as powerhouses of the cell, as ATP are formed inside this mitochondria. So what happens here? They generate ATP by oxidizing the selected food materials in the presence of molecular oxygen. So regarding the structure, they look like a rod-shaped structure, and they have double membrane. The outer membrane is smooth, and it is uh, linked with a number of spherical particles which contain NAD-linked dehydrogenases. The outer membrane is linked with NAD-linked dehydrogenases, and the inner membrane is convoluted. You can see here; these are the convolutions. Okay. So these convolutions are known as cristae. So these convolutions, they project into the inner chamber, and the outer membrane and inner membrane is separated by the intermembranal space. This is the intermembranal space. Then uh, the inner chamber, you can see here, this is the space bounded by the inner membrane. It is called inner chamber, and it is filled with the matrix. See this here. This inner chamber is filled with the matrix. And the matrix contains dense granules, 70s type of ribosomes, and mitochondrial DNA. Okay, this is regarding the structure of mitochondria. And one more thing, on the cristae, that is the finger-like projections, we said that they contain a spherical particles ranging from 8 to 9 nanometer in diameter with a short stalk and a head. Okay, so these spherical particles are called elementary particles. You can see here clearly. See, These are elementary particles or F0, F1 particles or ATP synthetases, and they help in formation of ATPs. Okay? And we also saw about types of respiration. We said that if the respiration occurs in presence of oxygen, it is known as aerobic respiration. And here, complete oxidation of the respiratory substrate take place, and then products the carbon dioxide and water. And a lot of energy is released in this process. And the respiration where no oxygen is utilized is known as uh, anaerobic respiration, and here the end products are carbon dioxide and ethyl alcohol. Here incomplete oxidation occurs and very little energy is liberated. That is anaerobic respiration. So you can see here the glucose in the presence of oxygen is completely oxidized into carbon dioxide and water, and a lot of energy, that is 686 kilocalories of energy is liberated here. But here no oxygen, it occurs in the absence of oxygen, and this glucose is partially oxidized to ethyl alcohol and carbon dioxide, and a very little energy, that is only 56 kilocalories of energy is liberated. Okay, so aerobic respiration occurs in almost all the higher living cells, but here anaerobic respiration occurs only in microorganisms. Next, we saw about glycolysis. We said that glycolysis is the common phase in aerobic and anaerobic respiration. Glycolysis, breakdown of glucose into two pyruvic acid molecules, and it occurs in a cytoplasm. Okay, so glucose is a six-carbon compound, and it ultimately breaks down into three carbon compound, that is pyruvic acid molecules, and it occurs in cytoplasm. Okay, so we will have a quick glance about glycolysis. Initially, this glucose undergoes phosphorylation reaction. It takes one phosphate molecule from ATP and forms glucose 6-phosphate in the presence of hexokinase enzyme. Okay, you can see here. But ATP is utilized. ATP is converted to ADP. The phosphate molecule is taken by this glucose and it forms glucose 6 phosphate. This reaction occurs in the presence of hexokinase. The next step, the glucose 6 phosphate is isomerized to fructose 6 phosphate in the presence of phosphohexokinase. And now this fructose 6 phosphate again undergoes phosphorylation reaction. Here also one ATP is utilized. You can see here, you have to note that here the ATPs are utilized. One ATP here and the other ATP here. Okay, so fructose 1,6 bisphosphate formation takes place, and this takes place in the presence of phosphofructokinase. So till now two ATPs are utilized. Now this fructose 1,6 bisphosphate undergoes cleavage reaction in the presence of aldolase enzyme. This is a 6-carbon compound. It breaks down into two 3-carbon compounds. One is glyceraldehyde 3-phosphate and the other is dihydroxyacetone phosphate. This dihydroxyacetone phosphate does not undergo any further reactions, so it again isomerizes into glyceraldehyde 3-phosphate in the presence of triose phosphate isomerase enzyme. So now we have two molecules of glyceraldehyde 3-phosphate. One is formed originally and the other DHAP is, that is dihydroxyacetone phosphate, is isomerized to glyceraldehyde 3-phosphate. So two molecules of glyceraldehyde 3-phosphate we have. Okay, now this glyceraldehyde 3-phosphate undergoes um, oxidation reaction. Here uh, hydrogen ions are removed from this glyceraldehyde 3-phosphate, and these hydrogens are taken up by this NAD, which is an oxidizing agent, and itself undergoes a reduction, forming NADH. Okay, this is very important reaction. You have to note this. Glyceraldehyde 3-phosphate undergoes oxidation, releasing H plus ions, and these H plus ions are taken up by NAD, and this reaction occurs in the presence of dehydrogenase enzyme. Here NAD is the oxidizing agent, which itself undergoes reduction. It reduces to NADH. Here in this reaction, one inorganic phosphate molecule is observed from the cytoplasm, resulting in the formation of 1,3-bisphosphoglyceric acid. Always aldehydes on oxidation turns into acid. So here glyceraldehyde turns into phosphoglyceric acid. Okay, and one phosphate ion is taken up from the cytoplasm, inorganic phosphate. So 1,3-bisphosphoglyceric acid formation takes place. Now this 1,3-bisphosphoglyceric acid loses one phosphate ion. Dephosphorylation reaction takes place in the presence of phosphoglycerokinase, resulting in the formation of 3-phosphoglyceric acid. And this released phosphate ion is taken up by ADP, and it converts itself into ATP. So here the first ATP molecule formation takes place. Here one NADH is formed, and here one ATP formation we can see. Okay, so this 3-phosphoglyceric acid undergoes mutation, that is the phosphate phosphorus group from the third carbon moves to second carbon, 2-phosphoglyceric acid, and this reaction is mutation, and it takes place in the presence of mutase enzyme. Now this 2-phosphoglyceric acid, it undergoes dehydration, where one water molecule is removed from this 2-phosphoglyceric acid, and this takes place in the presence of enolase enzyme and Ng2 plus ions acts as coenzymes, okay, cofactors. Now this 2-phosphoglyceric acid, on dehydration, it forms phosphoenol pyruvic acid, and in the last step it undergoes one more dephosphorylation reaction, where one more phosphate ion is liberated, and this phosphate molecule is absorbed by ADP and it forms into ATP. So as a whole, two ATP molecules formation we can see here, and this phosphoenol pyruvic acid turns to pyruvic acid, which is a 3-carbon compound, and this reaction takes place in the presence of pyruvate kinase. Okay, so this is very important glycolysis. Okay, so as a whole, total ATP formed 4. How 4? For each glyceraldehyde, 2 ATP. For each glyceraldehyde, 1 NADH. We have 2 glyceraldehyde molecules. One from DHAP and the other from glyceraldehyde. So each glyceraldehyde, 2 ATP. Total ATP, 4. Each glyceraldehyde, 3-phosphate, 1 NADPH. On oxidation of glyceraldehyde 3-phosphate, formation of 1 NADH. Okay, so here for 2 molecules we have 2 NADH molecules. So as a whole, ATP, total ATP formed 4, but already we use 2 ATP molecules, where from glucose to glucose 6-phosphate 1 ATP is utilized, and from fructose 6-phosphate to fructose 1,6-bisphosphate 1 ATP. So net gain is 2, and net gain of NADH is 2. You have to remember this. Okay, so we will enter into today's lesson. Okay. What happens to the pyruvic acid? Pyruvic acid cannot enter into Krebs cycle directly. So where does it go? It enters into the mitochondrial matrix with the help of a protein called translocator protein or permease protein. Okay? Permease or translocator. The end product of glycolysis, the pyruvic acid, enters into mitochondria from cytoplasm, and its entry is helped by a carrier protein enzyme called permease or translocator. Okay, so here this reaction, this oxidative decarboxylation of pyruvic acid reaction is termed as a link reaction because it links glycolysis and Krebs cycle. It is a connecting link between glycolysis and Krebs cycle. So here a small fraction of energy present in glucose molecule is liberated by the time pyruvic acid is formed, and the rest of the energy is liberated during the oxidation of this pyruvic acid into carbon dioxide and water in the presence of atmospheric oxygen. Okay, so here pyruvic acid directly enters into the mitochondrial matrix and is converted into acetyl coenzyme A. Acetyl coenzyme A. In this process, a pyruvic dehydrogenase enzyme catalyzes this reaction, and this pyruvic dehydrogenase enzyme is a complex enzyme with 3 enzymes, that is pyruvate decarboxylase enzyme, lipoate acetyl transferase enzyme, and lipoate dehydrogenase enzyme. These three enzymes together constitute pyruvic dehydrogenase complex. And also here you can see five coenzymes are very essential for these reactions to occur. What are those five essential coenzymes? Thiamine pyrophosphate, lipoic acid, nicotinamide adenine dinucleotide, coenzyme A, and magnesium 2 plus ions are the five essential coenzymes which are required for the oxidative decarboxylation of pyruvic acid into acetyl coenzyme A. And this process takes place in two steps. First step is the decarboxylation, and the second step is dehydrogenation. Dehydrogenation is nothing but oxidation. Okay, removal of hydrogens or electrons is a oxidation process. So in the first step, the pyruvic acid in the presence of TPP, that is thiamine pyrophosphate, and in the presence of pyruvate decarboxylase enzyme, turns into hydroxyethyl thiamine pyrophosphate. And in this reaction, one carbon is liberated in the form of carbon dioxide. So this is the decarboxylation reaction. In pyruvic acid, how many carbons are there? Three carbons. This is a three carbon compound. Out of three carbons, one carbon is removed in the form of carbon dioxide. So we are left with only two carbons. Okay, hydroxyethyl thiamine pyrophosphate is the substance which is formed by pyruvic acid on decarboxylation reaction. So here the carbon dioxide is liberated, and this reaction takes place in the presence of pyruvate decarboxylase enzyme. Now, in the second reaction, this hydroxyethyl thiamine pyrophosphate, in the presence of lipoic acid, it forms into acetyl lipoic acid, and here the thiamine pyrophosphate is liberated again. Okay, then there ends the decarboxylation reaction by the formation of this acetyl lipoic acid. And this reaction takes place in the presence of lipoic acetyl transferase enzyme. Okay. Very simple. See, oxidative decarboxylation of pyruvic acid. Here it undergoes oxidation as well as decarboxylation. It has three carbons. By the process of decarboxylation, one carbon is removed. So only two carbon compound will get at the end of this oxidative decarboxylation. So already in the first step, decarboxylation occur, and as a result, acetyl lipoic acid is formed, and TPP, which is utilized in the first step, is liberated again. Okay, so it is released. Now dehydrogenation reaction. Okay, so here acetyl lipoic acid, it in the presence of coenzyme A, forms into acetyl coenzyme A, and the lipoic acid is released in this state. Okay, so this takes place in the presence of lipoate dehydrogenase enzyme. Lipoate dehydrogenase, and this lipoic acid FAD acts as an oxidizing agent, and in the presence of this FAD, lipoic acid loses hydrogen ions, and FADH2 formation takes place. Okay, this FADH2 flavin adenine dinucleotide is a reduced form, and it's stored energy. And this FADH2 transfers the H2 uh, ions to NAD, and ultimately NADH plus H plus formation takes place. So as a result of oxidative decarboxylation of pyruvic acid, we will get acetyl coenzyme A plus NADH. Okay, for each molecule of pyruvic acid, we have two molecules as a result of glycolysis. Formation of two pyruvic acids molecules takes place. So each pyruvic acid converts itself into acetyl coenzyme A, and in this process, one NADH plus H plus is formed. So for two pyruvic acid molecules, two acetyl coenzyme A's plus two NADH. Okay, so you have to remember that here two NADH plus H plus uh, molecules are formed. Okay, so now this acetyl coenzyme A enters into Krebs cycle. Once again, pyruvic acid doesn't enter directly into the Krebs cycle. It undergoes oxidative decarboxylation, and as a result, see, oxidative decarboxylation in the presence of NAD plus coenzyme A and water, resulting in the formation of acetyl coenzyme A, and in this process, NAD is reduced to NADH and carbon dioxide is liberated. This is the whole reaction. Okay, pyruvic acid converts into acetyl coenzyme and carbon dioxide is liberated. This takes place in the presence of a complex enzyme called pyruvic dehydrogenase, which in turn contains three enzymes: pyruvate decarboxylate, lipoate acetyl transferase, and lipoate dehydrogenase. And this process takes place in two steps: decarboxylation reaction and dehydrogenation reaction. Ultimately, acetyl coenzyme and NADH formation takes place. Now, this acetyl coenzyme is ready to enter into Krebs cycle, and Krebs cycle takes place in mitochondria membrane. Mitochondria. So, mm, the sequence of reactions in Krebs cycle are described by Sir Hans Krebs in 1937. So, the cycle is known as Krebs cycle. And it is also known as TCA, tricarboxylic acid cycle. Why it is called tricarboxylic acid cycle? Because the first formed compounds, the first formed four compounds, that is citric acid, cis-aconitic acid, isocitric acid, and oxalosuccinic acid. All these are tricarboxylic acids. Okay, citric acid, isocitric acid, and cis-aconic acid. These are tricarboxylic acids. So we also call this cycle as tricarboxylic acid cycle, and it is also known as citric acid cycle because the first formed stable compound is citric acid. Here, six carbon compound, it's a six carbon compound, so it's also known as citric acid cycle. Here in Krebs cycle, it results in the generation of 3 NADH. You can see here one NADH here, one NADH, and one NADH here. As a result of Krebs cycle, 3 NADH and one FADH formation takes place, and one ATP. For each acetyl coenzyme A, we have two acetyl coenzyme A molecules, so we have to double this. 3 into 2, 6. 1 into 2, 2, and 1 into 2, 2. So 6 NADH, 2 FADH, and 2 ATP are formed as a result of Krebs cycle. Okay. Then, so we will see how it occurs. Okay. In the first step, this acetyl coenzyme as it enters into the mitochondria or mitochondrial matrix, the Krebs cycle takes place in mitochondrial matrix. So this acetyl coenzyme it enters into the Krebs cycle. It is accepted by oxaloacetic acid, which is a 4-carbon compound. This is a 4-carbon compound. This is the primary acceptor of acetyl coenzyme A, and this reaction takes place in the presence of citric synthetase. Here 2 carbons, here 4 carbons. As a result, a 6-carbon compound is formed. This is citric acid. Okay, so here coenzyme A is liberated from acetyl coenzyme. Okay. Acetyl coenzyme A plus OAA, oxaloacetic acid, which is a 4-carbon compound plus 2-carbon compound in the presence of citric synthetase, results in the formation of citric acid. Here coenzyme A is liberated. Okay. Then in the next reaction, this citric acid undergoes dehydration, that is removal of a water molecule, resulting in the formation of cis-aconitic acid, and this reaction takes place in the presence of enzyme called aconitase. In the presence of aconitase enzyme, citric acid undergoes dehydration, and in the next step, in the very next step, this cis-aconitic acid undergoes again a hydration reaction. That is, one water molecule is removed, and here again one water molecule will be added to this cis-aconic acid in the presence of aconitase enzyme, resulting in the formation of isocitric acid. Okay, so this isocitric acid undergoes first oxidation reaction. So, what is the aim of Krebs cycle? We have to the respiratory substrate has to be completely oxidized, okay? And the carbons, whatever are fixed in the respiratory substrate, they has to be released in the form of carbon dioxide. Okay, so here in this reaction, this is the first oxidation reaction in Krebs cycle, where isocitric acid in the presence of isocitric dehydrogenase, in the presence of isocitric dehydrogenase, and in the presence of oxidizing agent as a coenzyme. NAD is an oxidizing agent and it acts as a coenzyme here. So in the presence of isocitric dehydrogenase, this isocitric acid here undergoes oxidation and resultant product formed is oxalosuccinic acid. Oxalosuccinic acid. It is also a 6-carbon compound. So here, whatever H plus ions are liberated, these H plus ions are accepted by this NADH and it turns into a reduced form, that is NADH plus H plus ion. So you have to note that this is the first NADH formed in Krebs cycle, and this is the first oxidation reaction which occurs between isocitric acid and oxalosuccinic acid. So isocitric acid and oxidation gives oxalosuccinic acid. In this reaction, one NADH is formed. In the next reaction, this oxalosuccinic acid undergoes decarboxylation. What is decarboxylation? Removal of carbon in the form of carbon dioxide. Okay, so we have 6 carbons here in the oxalosuccinic acid. So one of the carbon is removed in the form of carbon dioxide, and this reaction takes place in the presence of oxalosuccinic decarboxylase enzyme. So this is 6 carbon and this is a 5 carbon compound which is formed as a result of decarboxylation of oxalosuccinic acid, and this compound is alpha ketoglutaric acid, which is a precursor for many of the amino acids and lipid substances. This is an intermediate substance which is precursor for many other amino acids. So this is very, very important. It is a 5 carbon compound formed as a result of decarboxylation of oxalosuccinic acid. Okay, so this alpha ketoglutaric acid undergoes one more oxidation reaction, oxidative decarboxylation. Okay, this is one oxidative decarboxylation reaction. Here, the second oxidative decarboxylation reaction. This alpha ketoglutaric acid undergoes, okay, in the presence of alpha ketoglutaric dehydrogenase. Here also NAD plus acts as a coenzyme. This is an oxidizing agent. It oxidizes in the presence of this NAD plus it undergoes oxidation. That is, this NAD plus draws the hydrogen atoms from alpha ketoglutaric acid. So these hydrogen ions are given to this NADS, and this NAD itself undergoes reduction, resulting in the formation of NADH plus H plus. So you can see that up to now we have 2 NADH formed. So in this reaction takes place in the presence of coenzyme A, alpha ketoglutaric acid plus coenzyme A gives rise to succinyl coenzyme A, and in this process carbon dioxide is liberated or released. So as it is an oxidative decarboxylation reaction, first oxidation, removal of H plus ions, decarboxylation, removal of CO2. These H plus ions which are removed from alpha ketoglutaric acid are accepted by NAD plus, and this NAD plus is reduced to NADH plus H plus. Okay, so this 5 carbon compound now, by losing one carbon in the form of carbon dioxide and reacting with this coenzyme A, it forms succinyl coenzyme A. Succinyl coenzyme A. Okay, so this succinyl coenzyme A, what happens to this succinyl coenzyme A? Here, this succinyl coenzyme in the presence of succinic thiokinase, it loses this coenzyme and forms succinic acid. Okay? In the presence of succinic thiokinase enzyme, this succinyl coenzyme A is converted to succinic acid, and this coenzyme A by succinyl coenzyme A synthetase, in the presence of succinyl coenzyme A synthetase or succinic thiokinase. Okay? And in this reaction, the energy of the thioester bond of succinyl coenzyme A is used to convert ADP into ATP. See here, ADP, in the presence of an inorganic phosphate, it accepts or it takes up the energy, whatever is released by breaking the bond between succinyl and coenzyme E. Okay, so uh, this energy is utilized for this phosphorylation, formation of ATP from ADP and inorganic phosphate. So this is known as substrate level phosphorylation. Here you have to remember this. In glycolysis also we saw substrate level phosphorylation, and in Krebs cycle also, in between succinyl coenzyme and succinic acid, there is a substrate level phosphorylation, ATP formation, by utilizing the energy which is released by breaking of bonds between succinyl and coenzyme. That is thioester bond of succinyl and coenzyme A. As a result, succinic acid formation takes place. Okay. And, up to now, we saw two oxidation reactions. Now, this succinic acid undergoes the third oxidation reaction. In this reaction, succinic acid is oxidized to fumaric acid. Succinic acid is oxidized to fumaric acid, and this reaction is catalyzed by succinic dehydrogenase. Succinic dehydrogenase. Here, this is also oxidation reaction. So H plus ions are removed from the succinic acid, and these H plus ions are accepted by FAD, flavin adenine dinucleotide, which is a coenzyme and oxidizing agent here. Okay, so it is the oxidizing agent, and it accepts the H plus ions from the succinic acid, and FAD is converted to FADH2. This is the third oxidation reaction, and this succinic acid forms into fumaric acid. And in the next reaction, this fumaric acid reacts with a water molecule to form malic acid, and this reaction takes place in the presence of fumarescence. Hydration reaction. This is a hydration reaction. In the presence of a water molecule, fumaric acid forms into a malic acid. This reaction is catalyzed by fumarase. And the last step and the final oxidation reaction. Until now, three oxidation reactions. One here, and one here. So this is the fourth oxidation reaction. Completely four oxidation reactions takes place in Krebs cycle. You have to know that. Okay. So in the last oxidation reaction, the malic acid is this malic acid is oxidized to oxaloacetic acid in the presence of malic dehydrogenase. And in this reaction, one NAD plus. See, this is an oxidation reaction. Oxidation is nothing but removal of H plus ions. So these H plus ions are accepted by this NAD, and it converts into NADH plus H plus. Okay, thus oxaloacetic acid utilized in the beginning. See, it is utilized in the first reaction is regenerated, and again it is ready to accept one more acetyl coenzyme A molecule. Okay, so as a whole, four oxidation reactions takes place in Krebs cycle, and two decarboxylation reactions. You have to note it. Okay, two decarboxylation. We have two carbons in acetyl coenzyme A. So these two carbons are removed by the process of decarboxylation. Two decarboxylation. The first one is oxalosuccinic acid undergoes one decarboxylation reaction. This is a 6-carbon compound, and alpha ketoglutarate acid is a 5-carbon compound. And this alpha ketoglutarate acid, it again undergoes oxidative decarboxylation. Here, this 5-carbon compound turns into a 4-carbon succinyl coenzyme in the presence of coenzyme. So this is a 4-carbon compound. So oxaloacetic acid is a 4-carbon compound which is utilized initially, and these acetyl coenzyme, the carbons which are present in acetyl coenzyme A, are removed by the process of decarboxylation. So two decarboxylation reactions to remove two carbons from acetyl coenzyme. So from here on, only regeneration of OAA reactions takes place. Okay, so two more oxidation reactions where only H plus ions are removed, and which are accepted by FAD here between succinic acid and fumaric acid, and NADA between malic acid and oxaloacetic acid. So as a whole, we have 3 NADH and 1 FADH as a result of 4 oxidation reactions, and also 1 ATP is formed in the process of substrate level phosphorylation between succinyl coenzyme A and succinic acid. The thioester bond between succinyl and coenzyme A breaks down, and as a result, succinic acid formation takes place. And the energy which is liberated as a result of this breakdown of thioester bond is accepted by ADP and inorganic phosphate. They bond together, resulting in the formation of ATP, and this is known as substrate level of phosphorylation. Okay, so now as a result of Krebs cycle, we have 3 NADH and 1 FADH and 1 ATP for each acetyl coenzyme A molecule. Altogether, we have 2 acetyl coenzyme molecules. So, 2 into 3 equal to 6 NADH, 2 into 1 FADH equal to 2 FADH, and 2 into 1 ATP equal to 2 ATP. So, as a result of Krebs cycle, we have 6 NADH, 2 FADH, and 2 ATP. Okay, all these are they undergo um, they are transferred through many electron carriers in electron transport system in the presence of oxygen. Ultimately, these H plus ions are given to oxygen, resulting in the formation of H2O, and a lot of energy is released, and this energy is stored in the form of ATP. Okay. Now we will see the importance of this Krebs cycle, that is significance of Krebs cycle. Okay. Oh. So this tricarboxylic acid cycle or citric acid cycle or Krebs cycle plays a major role um, as a common metabolic pathway to several other synthetic metabolic pathways like biosynthesis of lipids, proteins, and carbohydrates take place by using these intermediate compounds as precursors for the biosynthesis of lipids and other proteins. Okay, so some of the organic acids like pyruvate, oxaloacetate, ketoglutarate are capable of generating alanine, aspartate, and glutamate type of amino acids. Okay, so pyruvate, oxaloacetate, and glutarate, that is alpha ketogluteric acid, pyruvic acid, and this oxaloacetic acid are used as precursors for alanine, aspartate, and glutamate type of amino acids. Okay. And the main purpose of the cycle is to convert the potential chemical energy into ATP. So, whatever energy is stored in the respiratory substrate, that is initially it was glycolysis, now it is pyruvic acid, and which is converted to acetyl coenzyme A. So, that energy is to be converted into kinetic energy. The potential energy has to be converted to kinetic energy. Okay, so this cycle provides the reducing equivalents, hydrogens and electrons in the form of NADH2, FADH2 molecules, okay, which can form high energy ATPs by oxidative phosphorylation in the final step, that is electron transport system. So, and one more thing is that this Krebs cycle is an amphibolic process. You have to remember this is important multiple choice question which can be asked in your entrance examinations. It is an amphibolic process. Why it is called an amphibolic process? Because it includes both anabolic cycles and catabolic cycle. Catabolism. What is catabolic breakdown? Breakdown of the respiratory substrate, that is glucose first breaks down into pyruvic acid, which is break down into acetyl coenzyme A, and this acetyl coenzyme A ultimately undergoes decarboxylation, resulting in the formation of carbon dioxide. So this is the breakdown, and in the same way this is also an anabolic process serving as a central pool supplying the primers for the synthesis of various lipids, proteins, and carbohydrates. So it is both anabolic and catabolic process. So it is known as amphibolic in nature. This is all about today's class. So in our next class, we will see um, electron transport system. Thank you.